What I'm going to do in the first part of this, I'll be going through very quickly because this is marker material. We need to say why we want to work on aluminum, why I wanted to work on aluminum toxicity. Well, roughly 40% of the Earth's surface is acid. And as soils degrade, they become more acid. And you can see in this slide from Carlos Camargo in Brazil, you can get wheat that grows well on acid soils and that's not. And it's mainly affected by, in acid soils, you're limited with root growth. You're limited with root growth, the plants can die if it's too acid. This is a map. This is a really poor map. It's a USDA map showing acidity around the world. And you can see the major regions in developing countries that we need to work with acidity. Acidity is just as important in many regions of the world as salinity. We can solve acid problems two ways, by liming, which means farmers add lime to the soil, but this is not permanent. You have to add lime. Hi, Ronnie. Well, I saw this glow of light out there, and it was off the top of Ronnie's head. Liming. No, I'm not. A side joke. My wife gave me a birthday card last year that pictured an old guy standing there in his boxer shorts, kind of looking like this. And I'd opened it up and it said, you know you're getting old when you've got more hair in your butt than you do your head. Which says something about being married to a lady for 40 years. So liming is temporary. Varieties are permanent. So we wanted to look at producing more varieties. If you look in cereals, the most tolerant cereal in the world of the grasses is rye. The most tolerant wheat is about 32 parts per million, whereas Simmet has got ryes that are tolerant to maybe 200, 250 parts per million aluminum. So we developed a mapping population, recombinant inbred line, 150 lines. We used any kind of marker we could find, from AFLP markers to RFLP markers, and we're able to produce a fairly nice linkage map of about 2 centimorgans spanning the region of aluminum tolerance. Now, if we were plant breeders, the lecture's over. Because if I've got markers as a plant breeder within 2 or 3 centimorgans of my gene of interest, 95+% of the time, when I select for those markers, I'm going to get my gene. So, you know, you grow plants in the field, 95 out of 100 are going to have what you want. However, when you have students, we wanted to go back in and see how close we could get to that gene and see if we could clone that gene. Okay? 150 line mapping population was just not big enough for high resolution mapping. So we had to go back and start over again and build a 2,000 line F2 mapping population. So if any of you in this room are designing a population, make it as big as you can humanly possibly make it, because you never know if you're going to need it. We decided to go in and look at using rice and barley and wheat to map in rye. We were lucky enough to find out that there was an old barley cDNA clone, I think that's yours, isn't it, BCD 1230, that was developed here at Cornell, which is an epimerase gene and has nothing to do with aluminum. But we found it basically co-segregated with the ALT3 locus. We weren't getting enough markers out of wheat. We decided to look at the rice back library and see if we could find a rice back which contained BCD 1230. So we got a rice back which contained BCD 1230, developed primers from one end, put it on the map, develop primers from the other end, and put it on the map. And we knew then that these are the 2 primers at the end of the back, that in between these 2 primers contained the genes that we were interested in. So the next step was just to go into that rice back and make primers and start walking your way back in till you came to your gene of interest. And the techniques we were using in those days, you could see it was very easy to pick out what was segregating and what was not. I'm fast forwarding to the sequencing bit because I know this is what Leon wants to hear. So we walked back in and looked at all the genes, and we found a region that flanked our aluminum locus, and there were 5 genes in that region, 4 genes in that region. These 2 genes were not functioning, obviously had no effect on aluminum. This gene, I can't remember what it is, but it was nothing to do with aluminum. This gene was a transcription factor, class 3 trithorax containing transcription factor that only was expressed in roots, only in roots of a tolerant variety, and only when that tolerant variety was under stress. So it was not constitutive, it was induced. About that, we tried to go in and do map-based cloning to get at this gene, made primers from outside the gene, and tried to walk our way back in, and after 3 years, map-based cloning just was not in our possibility. About that same time, Sasaki in Japan published in 2004 on the ALMT locus in wheat. Malate transporter is a membrane transporter. Very important. But it is constitutive. It's always expressed in wheat roots, in rye roots, but the expression ramps up when these roots are under stress. What we had was not related to that. It was quite different, and it was induced. Malate, when you put a plant root, this is from Manny Delhousie, when you put a plant root under aluminum stress and you have free radicals of aluminum out in the environment, the root exudes malate through its membranes, the malate binds to the aluminum, and the aluminum can't get in. We were more interested in exclusion. There's several different kinds of aluminum. You can uptake the aluminum and sequester it, or you can exclude it. This appears to be an exclusion mechanism. At that time, with this high-resolution map, we found not only was our transcription factor located in this rye segment, but Nick Collins from Adelaide found that the malate transporter was also located adjacent to this transcription factor. We could not find that in rice because the malate transporter in rice is on another chromosome. Not only is it on another back, but it's on another chromosome. So the B26 transcription factor in rice is over here, the malate's over here. In the rye that we were looking at, they were right side by side. So we couldn't get at rye because we couldn't sequence rye. Rye, as you can see, is an absolutely massive genome. It's not as big as wheat, but compared to humans, it's about 4 or 5 times the size of a human genome. And rye has about 80% repeated sequences, and so trying to sequence a species where you've got 85 to 90% of the DNA is repeated, and it's the same repeat. So there's just no way we could put it together and assemble it. So I talked Peter Langridge into making a rye back library, and in Adelaide made a rye back library with the thought in mind we would screen the rye back library for a back that contained our region of interest and try and sequence a small piece of the rye genome instead of the whole thing. So Nick Collins and Bujung Shi in Adelaide screened the rye, screened about 5,000 backs, and found several backs that appeared to span this region. Now this is new. This is new information that hasn't been published, but what we decided to do was to go in and sequence 3 backs, and according to the screening, for example, this back contains at least 2 malate transporters and at least 2 B26 genes, and then a flanking gene. This one contains 2, 4, etc. The PCR primers we used flanking the malate transporter gene was telling us there's a lot of malate transporters in rye. So Olin, with the USDA-ARS, said, I'll sequence it back for you. Biggest mistake he's made in his career. We started out with 190 kb back, and after 2 years, he gave us 145 kb a sequence. With these gaps. All repeated sequence here, repeated sequence here. All of this was repeated sequence. There was nothing in it. But in this piece, we found it was a gene-rich region. We found all the genes we were interested in. Here's the ends of the repeats, which show here. Here's B26, which was what we were interested in. And here's malate transporters. So we decided to look at the malate transporter and compare it to Sasaki's. And we found that it had 6 exons, some introns, and they were in this order, exactly the same as Sasaki's indicated in the wheat system. This piece was a fragment, and this was most interesting. Throughout all the backs that we sequenced, we found, and I'll show you this again, we found either exons 5 and 6 or exons 1, 2, and 3. We never found exon 4 in a fragment. And these fragments, comparing sequence of the real gene versus the fragment, they were exactly the same. There's very little variation. So obviously, this gene can be broken into little pieces, and for some reason, these pieces are kept. Obviously, this is not functional, but it's still there, and for some reason, the PCR primers that we were using to identify these genes were identifying not only the whole gene, but identifying the fragments. I have no idea why they're doing that. Matoda in 2007 showed this is what the malate transporter looks like in wheat. This is outside the cell, inside the cell. We decided to compare the structure of the product of the gene and not the gene. As you well know, you can get a lot of variation within a sequence and you still end up with the same product. So when we looked at the malate transporter in rye, the sequence produced exactly the same product in exactly the same manner as Matoda said was in wheat. So from that, we conclude that the malate transporter in rye is the same as in wheat. This copy of the malate transporter. Then, at PAG in San Diego, Olin likes to drink margaritas. So after about 5 margaritas at a nice Mexican restaurant, he agreed to sequence another back. And then, as soon as he sobered up, he said, no, I'm not going to do it. But we talked Sandy Clifton, who was then at Washington University Sequencing Center, after a bottle of wine at the banquet, to sequence 2 backs for us. We started out with 190 kb back, and we ended up with about 130, 140 kb of sequence. So again, the repeated sequences make it very difficult to get. Well, you can sequence rye, but you just can't assemble it. We found 2 pieces that contain a malate transporter. One of them contained exactly the same as in the other back. And what you have here is you compare, that didn't come out very well, did it? You compare the exons are the same, little variation, but generally the same. So obviously, this other back that Sandy sequenced, which is not in the same location as the previous one that Olin did, this transporter is in multiple copies throughout the rye genome, because we've got at least 2 copies at 2 different locations. The fragment contained exons 1, 2, and 3, but in reverse, and the initiation codon was missing. So obviously, this gene can be broken into pieces, and the pieces still stay in rye. And again, the primers we used to identify this also picked out this, so which left me not too enthused with primers for identifying exactly what you have in what location you have it. So we did a third one, and for some strange reason, Sandy agreed to do another one. We found one malate transporter in this entire region, which said there was supposed to be 4, and there was only one complete copy. But this complete copy had lots of changes, as you can see. The intron was greatly expanded, had a chunk in it. This intron was missing. There was a mite inside this, which expanded it. So we doubt if this is functional. Yeah, there's a stowaway mite in the middle of this exon. So Sandy refused to do any more. Olin absolutely refused to do any more. But Dave Edwards from Australia said, yes, I'll do it, but we want to use 454 sequencing, which was a disaster. After a year and a half, he came back. Well, we got the genes, we got the information we needed, but he couldn't symbol it. 454, according to what I've been told, sequencing gives you a whole lot of little pieces, and then you have to put them together. Because there's so much of this repeat throughout the rye genome, after a year and a half, we had 98, 99 contigs, and he couldn't assemble them. But then I didn't care, because we could look within these contigs and find aluminum regions. Again, one of them, we found the complete gene, exactly the same as it was in the first 2 backs. We found a piece. Remember I said the pieces either contain exons 1, 2, and 3, or 5 and 6. We never found exon 4 in a piece. We found another one in another contig. In one contig, we found 2 malate transporters in tandem. So, we looked at this back, this one, and this one. This is the one that was 454 sequencing. This one we found 4 apparently functional malate transporters and a whole bunch of pieces. And we did not find any B26 or the transcription factor in there. We have no idea what the primers were picking up. Here we found lots of fragments, no functional malate transporter. Here we found 1, and here we found 1. So when we compared all these together with Olin's sequence, Albany sequence, there's 1, 2. This one we don't think in the Albany sequence was functional, and we found 5, or Dave Edwards found 5 in the Australian. Now, 454 sequencing gave me exactly the answer I wanted, but we couldn't put it together. We couldn't assemble it. So if you want to assemble a sequence, boy, at least in rye, I certainly would not use that methodology. Now let's digress off the malate to what I thought was interesting, this gene-rich region. Rice and rye have been separated by, what, 60 million years, roughly? Something like that. These 3 genes were in exactly the same order in rice and in rye, and exactly, well, very, very close to being the same distance from each other. We could not look at the rest of this. Well, Leon works for the ARS. We ran out of money, we ran out of time, we ran out of everything. But I would love to have, we have the sequences for all this. If somebody would like to go look at the collinearity between the grasses, because now we can do wheat, rice, barley, oats, brachopodium. We could take these genes and see if this particular region is highly conserved throughout the cereals. So now I'm going to change and look at the B26, the transcription factor, and see what we have. As we said, it's induced. We don't know if it works with aluminum. And I'll explain how Leon's going to solve that problem for us. Because all we know is that it's a transcription factor. It's induced in root tips and in root tips of tolerant varieties when they're under stress. So we looked at this transcription factor in the first back. Here we have 100, this is a sequence comparing rye and rice. This transcription factor is also in rice. And we haven't tested rice for it's induced under stress or not. But here's the sequence. There's the set domain. We assume, according to the sequence, that this is 100%, that this is a functional transcription factor. When you look at the other three fragments, we contain from 31% to 67% of the transcription factor. So these are pieces. You go into the Australian and look at their backs. You can again find pieces, but you don't find anything that's functional. Lots of pieces. But as far as we've been aware, after screening 5,000 backs, we've only found one, what appears to be completely functional transcription factor. How this relates to the upregulation of the malate transporters, we don't know. However, Leon's going to solve this problem for us. The biggest problem in the past is you can't tissue culture rye. You can't take it down to a single cell suspension, therefore you can't do transformation on it. And you sure if you can get it down close to a single cell suspension, it won't go back up or regrow. However, Leon's going to make triticales out of these ryes because for some reason the rye incompatibility to tissue culture and cell suspension is turned off when put in a wheat background. So we've got the sequences, so I can see 4 constructs to answer all of our research. One construct with no insert, one construct with B26, one construct with the malate transporter, and one construct with them both together in a triticale that's totally susceptible to aluminum. Straightforward, right, Leon?
Can you transform?
He's trying to get Mark involved in this. We found what appears to be a fully functional ALMT gene in Blanco rye. Blanco rye, the reason we use Blanco rye is it's a white, agronomically acceptable rye that's still grown widely under production in Brazil. Did I say it was a white rye? It's also a white seeded rye.
Blanco means white.
See, he works for the government.
Multilingual.
Multilingual. It appears that 6 exons are necessary for the function and that we found fragments all over the place. So if somebody wanted to tell me there were 10 copies of malate transporter in this Blanco rye, according to PCR analysis, you'd say yes. When you look at the sequence, you'd say no. There's only 4 maybe. Those 4 are located on at least 2 different chromosomes. Because one functional ALMT is on chromosome 3. The genetic region we were looking at is on the long arm of chromosome 4. So in Blanco rye, this gene, this region, throughout evolution, has appeared to be duplicated, moved around, whether jumping genes were involved or not, but it's moved around. If anybody that's looking at genome evolution of almost any species, they don't stay in the same place, they move. So far, gene collinearity, which is amazed me more than anything else, is 100% in this region. We have the sequences of the other 4 genes in that region, which would be nice if somebody would map it. We have the mapping population. Mark Sorrells has the ITME mapping population, so this could be done not only in rye but done in wheat to see what the collinearity is on this. There's at least one apparently functional transcription factor in Blanco rye, which is tightly linked to the malate transporter. The big question is, does this transcription factor that is induced and upregulated under aluminum stress, does it have anything to do with the expression change of malate transporter? Because the key to aluminum tolerance in rye is the membrane transporter exuding malate, which ties up the aluminum and prevents it from getting in the plant. So the first 3 minutes are what I would do if I were a plant breeder. The last 3 is trying to understand the biology of what you're working with. This is a few of the people that worked on this project over many years, starting with Andrzej Aniosz, a physiologist in Radzikow in Poland. I first met him in '75, and he was screening Polish rye for aluminum tolerance, and he said, look, I can do this in a bucket in the lab. And he's the one that started it. Carlos Camargo and Augusto Bayer in Brazil, they're the ones that said, the lab analysis doesn't mean a damn thing unless it correlates to the field. So we took hundreds of Brazilian varieties and compared their field data with exactly what we were doing screening in hydroponics. And the correlation was like 98%. The back library was a result of all these guys in the screening. Olin did 1, Sandy did 2, and then Dave Edwards did 1. So nothing like collaboration and cooperation. My blow time?
Yep.
Okay. If you work on a daily basis in Adelaide in the lab, that's what you wear. If the photographers come around for the lab newspaper, you have to scrounge around and find lab coats to put on to wear, because they refuse to take photographs like that. So there's a lot left that can be done. There's a lot of answers that we've got. There's a lot. The more we do, the more questions you have. And for those of you who are not aware, I assume all of you are, because Ronnie's in the room. Are you all aware of the Borlaug meeting next March in Mexico? Okay, everybody show up. But I will warn you, and I'm sure Ronnie's going to do this, the flight connections into Ciudad Obregón are horrible. If you don't get your flights booked really quickly, you're not going to be able to get into that city. There's was there, Ronnie, one flight a day from Mexico City?
No, I think there's a couple.
Couple flights.
Morning and evening.
Yeah. And the other worst case scenario is that the last week of March is spring break for most major U.S. universities. So Aeromexico's already doubled their ticket prices. What right today you could buy for $800 is online for $1,400 in March.
We're going to add flights.
You're going to add flights?
Seriously, the Senate has the...
USDA is going to...
Yeah. Oh, yeah.
Oh, yeah. USDA's putting in big bucks.
Yeah. Deep pockets.
Deep pocket.
USDA is hiring armored cars.
Yeah. So if you have, I'm open to questions. If you want to ask any, unfortunately, I volunteered to help organize the Borlaug event. So if you have any questions on next spring in Mexico, let me know. Any questions about rye?